How does stable isotope probing work? Stable isotope probing is an innovative MBT that uses biotrap amended with carbon-13 labeled contaminant, example carbon-13 benzene, to conclusively determine whether biodegradation has occurred. Because carbon in nature is almost exclusively carbon-12, carbon-13 is easy to track, as long as the compound is utilized by microorganisms as a carbon and energy source. The carbon-13 label serves much like a tracer which can be detected in the end products of biodegradation, microbial biomass, and CO2. So, how does it work? First, the desired compound is synthesized of carbon-13, like benzene shown here. It is loaded on biosep beads, which are made of powdered activated carbon and Nomex. The Nomex holds the pack together, like chicken wire. The pack sorbs the compound onto the bead, but the compound is still bioavailable to microbes. Carbon-13 compound is loaded onto beads at the lab. These loaded beads are then placed into a biotrap sampler. The biotrap is a passive sampling device that can be deployed in pre-existing monitoring wells. The biotrap is placed into the monitoring well for 30 to 60 days. Microbes that are naturally present in the groundwater will colonize the biosep beads and form biofilms. Organisms with the ability to degrade the labeled contaminant will begin to utilize it as a carbon and energy source. When they do, the heavy-weighted carbon will be incorporated into the new microbial biomass or metabolized as waste products like carbon dioxide. Carbon-13 is incorporated into the new cells growing within the beads or into the CO2 being produced. Biotrap samplers are retrieved from the monitoring well and shipped to the laboratory for analysis. At the lab, the remaining carbon-13 compound is measured. Since we know the original amount loaded onto the trap, we can calculate a relative rate of loss during the deployment period, and more importantly, we can look for the heavy-weighted carbon in the biomass and waste products. If we see unnaturally high amounts of carbon-13 in the biomass or CO2, it provides definitive proof that the organisms utilized our compound of interest. There's no way to have so much carbon-13 in these biomolecules unless the organisms used the label compound placed on the biotrap as a carbon and energy source. Proof positive that the compound of interest degraded during the deployment period. Contact us today to see how stable isotope probing can be utilized at your site, or check out more resources on our website, microbe.com/stable-isotope-probing.